Hi, my name is Peter Hollman. I'm a faculty member with the Department of Biological Sciences here at SFU. So in today's demonstration video, I'm going to introduce an important technique, the polymerase chain reaction, abbreviated PCR, and some of the associated equipment. So the polymerase chain reaction is a method that is used to make many copies of a specific sequence of DNA, and it relies on having a carefully designed specific primers that we use. Primers are specifically designed to only bind in specific places of the genome where this would not work. The students in our teaching labs, and you'll see one of them, use this equipment and get hands-on experience to learn this methodology. Developer of the technique won a Nobel Prize because it has been so important and changed the industry. It is used in all sorts of techniques such as cloning DNA and gene sequences, but it's also very useful for diagnostic assays and is one of is currently considered the gold standard to detect the coronavirus. In order to do this type of work, you have to be meticulously clean. Part of that is putting on PPE, personal protective equipment. The PPE helps keep contaminants out of our work, but it also protects us from some of the reagents we use. In order to copy DNA, you need all the ingredients for DNA. We include all those ingredients often together in something we call a master mix. So this includes the building blocks of DNA, deoxyribonucleotides with the different bases, A, G, C, and T, as well as a DNA polymerase, which makes the actual copies of the DNA. So the PCR ingredients are combined with the template DNA, which is the sample. So if you want to think about that in terms of a patient sample, if you wanted to find out if they had a virus, you would put in the patient's DNA sample, and then you could see if the viral nucleic acids are in the sample. Because the polymerase chain reaction depends on rapid temperature changes, we actually do it in an instrument called a thermocycler, which is capable of doing that very quickly. And so essentially, the PCR tubes are put in a block on the thermocycler. There is a program that's run, raises the temperature, drops it to specific temperatures, and then raises it again over many cycles. And this is really important because the technique depends on the double-stranded DNA being melted, essentially, denatured and becoming single-stranded, and allowing the specific primers to find their place in that single-stranded DNA. And so at a high temperature, the DNA becomes single-stranded, and then the temperature is lowered to allow what we call annealing of the primers. This is the specific binding of the primers and base pairing to the complementary DNA in the sequences. And then the extension step is done at a higher temperature by the thermostable polymerase, and it basically incorporates the nucleotides and makes the complementary strand of DNA. So essentially the polymerase is reading the template strand of DNA and extending the primer in one specific direction. After a round of PCR, if you had a single copy of DNA, you would have 2 copies of that template DNA. But if you did that again, you would then have 4, and then you would have 8. So it's an exponential increase, and over a period of about 35 cycles, you will have over 1 billion copies of that amplified DNA. And because the technique depends on these primers only being extended in one particular direction, the product, the amplicon that you wind up with, is a discrete sized piece of DNA that is basically flanked by primers at each end of the sequence, and that is what you wind up detecting and seeing. In order to detect the DNA from the PCR reaction, we run it in an agarose gel, and this is called gel electrophoresis. There are so many copies made of the amplicon that we can visually see it in a gel using an appropriate stain. We load the PCR product into the gel, as well as a reference pieces of DNA of known size that we include so that we can see what size the PCR product is. And so both these samples are loaded on the gel. Both the latter and the PCR product with the loading buffer are included in a lane on the gel. DNA is electrically charged. It has a negative charge. The negatively charged phosphate groups on there allow it to move towards the other end of the gel. And short pieces of DNA will travel much faster than big pieces of DNA. And so we can separate DNA of different sizes on a gel like this. The dye is gathered by the DNA as it travels through the gel. Eventually, we can see the DNA because the dye will fluoresce. In order to see the DNA on the gel, we have a machine that we call a gel documentation system. And essentially, it's a light source. It has filters that allow only the wavelength of light that you need for the fluorescent dye to fluoresce in order to image where the DNA is in the gel. So what we do is we take the gel, we put it onto the appropriate filter tray, and then we put it into the instrument. And essentially, if there is enough DNA gathered in a particular spot on the gel, it shows up as a fluorescent band because it has gathered the fluorescent dye. We can look at the gel, and if we see a band of the expected size, then the presence of a band of the expected size indicates that the sample tested positive. The PCR technique has been incredibly important in many areas, from forensics to biotechnology, genetic engineering, as well as diagnostics for detecting viruses.